So I'm going to share two ways that scientists have organized this fold universe to be able to make more sense of proteins. I'm going to show you the databases in a second, but I'll first show you a sneak peek just to get you to realize what we can use the databases for. This is a database called CAT, and I'm going to take you through the rings in a second, but the green ring here corresponds to really overall architectures that we can have proteins in, and the size of them corresponds to how common they are. So this is a class of some sort of orthogonal bundle where we would have lots of alpha helices, the globin folds too. We have the beta barrel folds, or architectures in this case. Some sort of beta sandwiches. We've looked at those too, right? Remember? You can have, instead of a one sandwich, you can have a two-layer sandwich. I don't think that's visible. We can have a three-layer sandwich. This is kind of the Rossmann fold, right? And then you can have some alpha and beta complex. There were a bunch of others that I ignored here, but you see that some of them are much larger than the others. So with only, say, 8 or so of these, I could represent the vast majority of all folds possible.